Dear students, now I am going to discuss gene regulation in eukaryotes. This video is in continuation with my previous. I discussed about promoter elements and enhancers. Today I am going to discuss about activators, repressors and silencers. So you know already gene regulation in eukaryotes in text space in different levels. It is chromatin or genome level, transcription level, transcriptional level, translational regulation, post translational regulation, hormonal regulation likewise. So here we are discussing only about transcriptional level regulation. We have discussed about how exactly the promoter elements regulate the gene expression. Similarly, what are the functions of the enhancers in gene regulations? We have discussed about this in our last video. So see this cartoon. So this office, there is a office or a firm is running. So several people, their rate of working pace is different. So that is what going to contribute to the production or the outcome of from that office. So their production rate may be in this rate, or else it may be sometimes it is going up and down like this. So sometimes it is slowly progressing, sometimes it is in the downward. So similarly, each gene expression will be like that. It is controlled by so many factors. So these so many factors, if they are contributing in unison, then you may see the gene, the production of the protein will be like this, you can see. So the gene expression in this space. If it is not in that pace, then you will be see like this. So this all this graph, or else it is your own progress in the college, or else in education, you may see all this. You can see there are so many factors. So it may be the factors in your home, or else in the college, or else in the teachers, or else the materials, the all these contributing towards your progress. So it different so that is what you can see, the gene regulation. So it is each cell, for example each cell, they have the composition of the genetic material will be the same, but the gene expression will varies because of these factors. For example, the skin cells are producing keratin, you can see. So these the intestine cells are not producing. So beta cells are producing insulin, but insulin producing gene is present in every cell in the body. So gene expression will be controlled by several factors, and you can see the rate of gene expression. So all these you can see, you will able to understand from this cartoon, I think. So this transcriptional controlling elements, what you see in eukaryote, that regulate gene expression by varying the rate of gene transcription or rate of mRNA production. So it is the gene expression means mRNA, then mRNA get translated in the ribosomes, that all those. So the rate of transcription initiation is the primary regulatory element in the gene expression. Gene for abundant proteins transcribed at very high rate. Genes for proteins required less, they are transcribed at lesser rate. For example, B cells produce antibodies in large quantities as per antigenic stimulus. So before the antigenic stimulus, B cells are not producing antibodies. So this how similarly I have given the example of the insulin, same thing. So the regulation of gene expression achieved by the interaction of promoters, transcription factors, enhancers, activators, silencers, repressors, so cis and trans acting elements. So all these you have see here. So the repressors will down regulate the transcription. So the activators enhancers also they increase the gene expression or transcription rate. So the eukaryotes protein coding genes are all transcribed by RNA polymerase 2. This RNA polymerase 2 binding and producing mRNA depends on all these factors. So the transcription is initiated by the formation of transcription initiation complex. That is what you will find here. That is, you can see with number of transcription factors, elements, so many elements, enhancers, silencers, repressors. So and also ultimately, so all these elements, they are influencing the transcription factors to bind with the Tata box, so that RNA polymerase is going to synthesize mRNA that is going to be translated in the ribosomes. So this is what you can see. There are so many controlling elements, what you see. So these transcriptional controlling elements, there are two types. Transacting, they are not the part of the DNA sequence. They are synthesized in the cytoplasm, then they are transported back to the nucleoplasm, and they are affecting the DNA sites and modulate the transcription of in the DNA. So those are called transacting elements. So these are the ones, the transcription factors and or activators and repressors. They are protein in nature. Then cis-acting elements, they are nothing but DNA sequences itself. They can only influence the expression of adjacent genes. So you will find promoter elements, enhancers, silencers, all these you will find here. So enhancers, so then the silencers, they are the part of the DNA itself. They are influencing the transcription. They are cis-acting elements. Transacting elements are, they are the activators, what you see here, transcription factors. So all these, then these are all the transacting elements or repressors. So again I can give all these factors, they are like so many people in an office, they are contributing to the amount of gene expression, amount of mRNA. All these depends on the activity of all these factors. So how exactly these factors, this is the DNA, just imagine. So the enhancer, so you will find this is the cis-acting, this is the part of the DNA. So how the enhancer affecting the promoter region? By producing activators, or these are called transacting elements. Through transacting elements, activators or transcription factors which are going to bind with the promoter sequence, so that the mRNA will be produced. So then how exactly the silencer, this also cis-acting, that is the part of the DNA. So this by producing or by activating repressors, so the repressor is going to bind the promoter sequence, so that the transcription is inhibited. So these are how, this is the basic model how they are going to control. Now let us go one by one, silencers, repressors and activators. Silencers, so you know that if there is no silencers, you cannot able to tolerate the the horn or the noise of this bike. That's why the silencers are adjusted, otherwise you will take fine from the traffic police. So the silencers are similar to the enhancers, cis-acting element, that is the part of the DNA sequence. They are located in the upstream or the downstream. The upstream means the sequence, DNA sequence, those are present the right side of the transcription start site, and the other one, the downstream, means they are in the left side. So the silencer affect the opposite. This is actually how the enhancer activates the transcription. Silencer, it is not, it is suppressing the transcription, or else it is slowing the transcription rate. So the silencer elements are decreasing the level of transcription. They contain binding site for repressive transcription factors or repressors. So you will see the part of the DNA. So it is having a binding site for repressors. So by binding to the repressors, it is going to contact the promoter and it decrease the transcription, or else it is stop the transcription. So the repressor transcription factors can either block the binding of an activator so that transcription is stopped, or else the silencer effects similar to those enhancer an independent orientation and located from a distance. From a distance it is affecting the transcription. Silencer modulate transcription from a distance. It may be present 1000 nucleotide or base pairs away from the transcription initiation site. When repressor binds to the silencer sequence away from the promoter, now once it binds, the repressor binds to the silencer, the DNA take a loop because it has to contact these the promoter area, right? So it DNA takes a loop so that repressor is in contact with the initiation complex. Repressor interacts with the transcription initiation complex and prevents the initiation of the transcription, so that the mRNA is not going to made. So in Drosophila there are two types of silencers, long range and short range. Short range silencers are very close to the promoters. Long range silencers are away from the promoters. They are affecting by taking a loop like this, you can see. Then what are the repressors? Repressors are opposite to the activators. Okay. Repressors, gene expression is regulated by repressors. They are nothing but they are transacting factors or else protein factors. Eukaryotic repressors, they bind these specific DNA sequences and inhibit the transcription. Transcription repressors can bind the promoter or enhancer region, so that block the transcription. So you will be able to see here, this is the promoter region, Tata box. So this repressor bind in this region so that activator cannot, or transcription factor cannot bind now. So transcription is not initiated. In some cases, eukaryotic repressors simply interfere with the binding of the transcription factor. That's what I said. The binding of the repressor near the transcription start site can block the interaction of RNA polymerase to the general transcription factor. There are a set of general transcription factors which is very important. So to recognize the RNA polymerase 2 will bind to the Tata promoter region, these transcription factors are important. So this interfere with that, so that transcription is not initiated. So repressor can either block the binding of the activator directly, compete with the same binding site. So it may bind, so where the activator going to bind, that binding site will be occupied by the repressor, so the activator cannot bind, so that transcription is not initiated. Repressor may compete with the activators for the binding specific regulatory sequence. Such repressor contains same DNA binding domain. So the activator ಗೆ ಯಾವುದು ಡೊಮೈನ್ ಇದೆಯೋ ಅದೇ ಡೊಮೈನ್ ಇರುತ್ತೆ, so that it cannot the activator cannot now make contact with the promoter, so inhibiting the transcription. So gene expression results when only maximum polypeptide is made in the cell, so that you can see once the gene expressed, I mean the maximum amount of protein is made or mRNA is made, now the repressor get activated and it stops the transcription process. So this is the many transcriptional repressor proteins remodel chromatin, so that the chromatin undergo coiling, and once the transcription, the histones going to bind and transcription going to stop. So this is how the repressor function, and it down regulate the transcription. Now coming to the important transcriptional controlling elements, they are called activators. So apart from the basal transcription factors, these are the basal transcription factors, they are very important for the binding of the RNA polymerase to the promoter sequence. So they are so apart from that, there are several general transcription factors. They are nothing but the proteins. they regulate the gene expressions. They are called trans-acting elements. So these are called transcription factors or activators and repressors. All these are trans-acting elements. That means they are synthesized in the cytoplasm and they are transported to the nucleoplasm, and they are going to bind the promoter region or DNA directly, and they are going to regulate the gene expression or mRNA production. There are more than 100 different proteins. They take part in the specific regulation of a particular gene. This is what I showed in the cartoon, remember. So these transcription activators, so all these are the activators, you can see the transcription activators. They bind the promoter region specifically, and they enhance, they are functioning, you can see the enhancer influence the transcription through these activators. So this is what you can see. So the enhancers, you can see the associate with the modulate the gene expression through these activators. Okay. So what happens in this cartoon I have shown here. So when these activators are not functioning, or else they are not activated by the enhancer, what happens? The amount of mRNA produced is only this much, or gene expression is this level. When all these enhancer activate all these activators, what happens? The amount of the RNA production, this is the bar shows the amount of mRNA produced or amount of the protein produced. So these activators or transcription factors, they are trans-acting factors. They have multiple domains. So the domain means the region which make contact with the DNA, or else the protein, that is called the domain. So these required, they are called DNA binding domain. Some factors, they have DNA some factors having transcription activating domain, some factor having ligand binding domain. Remember, there are hundreds of transcription factors. They are called activators. They are required for the transcription of different genes. Okay, so they have the domain. They may bind with the DNA binding domain. Then they may binding with the transcription factors, transcription activating domain, or else ligand. So they bind with the ligand, some ligand. So these are the three classes of transcription factors, they depending on their binding domain. First, you can see the DNA. All these activators are large family of proteins, regulate the protein coding genes. These are they are distinct from basal transcription factor. They are different from basal transcription factor. Okay, some of these transcription factors are activators, so occur specific cell types. Some will present in all cell types. So each of these associated with promoter to modulate the rate of transcription. They have multiple domain, that's what I said, DNA binding domain, transcription activating domain, or ligand domain. So these are different transcription factors. Let us study this. DNA binding domain recognize specific DNA sequences in the regulatory region of the gene. So these DNA binding domain transcription factors, they are common structural motifs. Motifs means like similar to enzyme active site, what you have studied. They are made out of some amino acid residues. You studied in last semester. Similar types of motifs. They are called helix turn helix motifs, zinc finger motifs, leucine ziffer motifs. So the DNA binding protein possessing these motifs bind the high affinity to their recognition site, thereby they modulate transcription. So these motifs interact with the DNA through hydrogen bonds or van der Waals force. So this is the binding of these transcription factor motifs. So these are all DNA binding transcription factors. They have three different motifs. Depending on that, they are classified. Okay. So these based on the presence of these common structural motifs, these proteins are classified. They are DNA binding transcription factors. Remember, helix turn helix motif. Helix turn helix motifs means they have two alpha helix, you can see. They also have a beta turn, you can see. So this is the structural motifs what you see in the transcription factors of DNA. These are the helix turn helix motifs. Okay, so see these helix turn helix motifs, so they bind with the one of the helix bind with the major groove. So they associate with the major groove and they modulate the transcription. Okay, so where you find what are the homeobox genes transcription factors, they have these homeobox genes are the which play important role in embryonic development differentiation. Okay, so those transcription factors, they have this helix turn helix motifs. Then similarly cell cycle controlling factors mic gene, so they you can see these transcription factor, they have this motif and they bind with the they make contact with the major groove of the DNA and modulate the transcription. Similarly zinc finger, so zinc finger motifs is the, they have a you can see the two-stranded anti-parallel beta sheet, you can see anti-parallel beta sheet, one is in this direction, another is in this direction. So then there is a alpha helix, you can see in this, and all these they are connected with a zinc ion. You will find a zinc ion, you will see here. So this is what you can see here. So this also, this alpha helix makes a sequence specific contact with the major groove, what you see. Zinc finger proteins are most abundant proteins. Their functions are extraordinarily diverse. They include DNA recognition, RNA packaging, transcriptional activation, regulation of apoptosis, protein folding, assembly, lipid binding, hormones receptors. So many transcription factors, they have zinc fingers as a DNA binding domain. DNA ಜೊತೆ ಬೈಂಡ್ ಆಗೋ ಡೊಮೈನಿಗೆ ಜಿಂಕ್ ಫಿಂಗರ್ ಇರುತ್ತೆ. ಸೊ ದಿಸ್ ಇಸ್ ಆ್ಯಂಡ್ ದೇ ಮೇಕ್ ಕಾಂಟ್ಯಾಕ್ಟ್ ವಿತ್ ದಿ ಮೇಜರ್ ಗ್ರೂಪ್. ಓಕೆ, ಸೊ ದಿಸ್ ಇಸ್ ವಾಟ್ ಯು ಕ್ಯಾನ್ ಸಿ ದ ಜಿಂಕ್ ಫಿಂಗರ್ ಡೊಮೈನ್. ದ ಲಾಸ್ಟ್ ಒನ್ ಈಸ್ ಲೂಸಿನ್ ಜಿಫರ್ protein. So these transcription they have a leucine residues. Leucine is a amino acid residues, is in the 7th position of the C terminal of the DNA binding domain. These protein using two extended alpha helix, you can see two extended alpha helix, and they bind with the DNA at two different major grooves. You can see here the one alpha helix to this groove, major groove, another to this. So they have a leucine zipper. So they have a you can see zipper like, you can see zipper, you know, zip. So this zipper protein motif have an amphipathic alpha helix at their carboxyl terminal. One side the helix consists leucine, that you can see every 7th position of the amino you will find a leucine residue. So this is you can see the charged polar groups, you can see, so that they make contact with this. So this basic amino acid interact with the phosphate backbone of the DNA, electrostatic interactions, you can see. So this is what you can see, the leucine zipper protein. So transcription factor. So apart from this, you can see transcription activating domain. Transcription activating domain enhance the transcription by physically interact with the regulatory protein. You have seen that. Then ligand binding domain. Ligand binding domain is how steroid hormone bringing the, you know, for example the androgen. So it is enhances that protein synthesis, muscle proteins. Okay, so this how exactly testosterone receptor, this once this testosterone bind to the receptor, this migrate or transported to the, so it directly make contact with the DNA and it enhances the transcription. So this is how they activate. So this is called ligand, this is the ligand binding domain. So this is about the activators and different transcription factors, how they turning on and turning off the gene and make you know the gene expression. Thank you.